We resume now with our previous lecture, looking at advanced topics in neurobiology and endocrinology, with the themes being informational stuff, exactly the same in the two systems. All the dogma you've learned already come with elaborations. It is not always the case one hormone causes one hormone. It is not always the case one neurotransmitter coming out of axon terminals. There's more complexity, multiple messengers, spatial control as to different branches of a neuron, as to what axonal branches will be having how much of an action potential, different branches coming out of the hypothalamus, all of that. We move now to the next version, the next elaboration, which is forever, as people began to learn about things like neurotransmitters and hormones, an incredibly dominating state of mind was to say, well, how much neurotransmitter is being released into the synapse? How much of some particular hormone is in the bloodstream? How much of a messenger is there? And both fields were dominated by this reasonable notion that you really need to know that to know how big an effect you were going to have on a target neuron or a target cell in the body, how much of a messenger. And what people began to realize only some decades ago with fancier molecular techniques is that's only half the story. How much of a signal are you releasing, and how much is the recipient of that signal listening? And you have here this world where you could be broadcasting by radio. Do people still broadcast with radio? But incredibly powerful radio waves going out all over the place. And what a strong signal. But if you happen not to have a radio, you have no ears, you do not receive the message. It is just as interesting to ask how much is a target neuron or cell in the body listening to a neurotransmitter message or a hormonal message as how much of a message is being released. And what this introduced people to was the notion that receptors, neurotransmitter receptors, hormone receptors, could change their number. The number of them could change. What came to be called autoregulation, much like the autoreceptors from neurons the other day, negative feedback loops. In this case, autoregulation. How does autoregulation work? We all know this. If somebody is constantly screaming at you, you stop listening to them. You down-regulate your sensitivity to what this person is yelling about. On the other hand, if it's important and somebody's whispering, you really get attuned to, you have up and down regulation in response to changing levels of signals to try to balance things out. And in the neurotransmitter realm, you see examples of this. You have a neuron is releasing tons and tons of neurotransmitter at presynaptic end. And in some circumstances, the postsynaptic neuron will downregulate its number of receptors, will decrease it somewhat just to get a little more equilibrium in the system, drop down the amount of neurotransmitter or hormone being released, and you will get a compensatory upregulation. Stuff like that changes. Where is that of interest in the neurobiological realm? We are going to see when we get to depression. There is a really straightforward explanation about the neurochemistry of depression involving the neurotransmitter serotonin. That is totally straightforward and explains everything, except it really does not explain a lot about what's going on, because there's some key confusing things you then see that could only be explained by a god-awful complicated scenario we're going to look at of postsynaptic neurons up and down regulating number of receptors. We will get to that. So the amount of serotonin receptors, equally important. At the endocrine end, you see an equivalent of this in a disease our great-great-great-great-grandparents never had to think about: adult onset diabetes, type 2 diabetes, insulin-resistant diabetes. Insulin, you secrete it from your pancreas. Don't worry if you're not on top of this. This is just give you a sense. You secrete insulin. from your pancreas when food hits the bloodstream. And what insulin does is, among other things, it tells your fat cells to take up glucose. Great. Your fat cells have insulin receptors and insulin binds to it, and as a result, glucose transporters activate and pull stuff into your fat cells. Suppose instead you are very, very overweight. Suppose your fat cells are completely full. They are jam-packed with every bit of energy they can store, and the fat cell, in a sense, says, screw this, we have no room for any more glucose, we're not taking up anymore, we are not going to listen to insulin anymore. We are going to remove our insulin receptors.
Insulin receptors are made by ribosomes that are attached to the membrane of the endoplasmic reticulum, or ER. Newly made insulin receptors fold and dimerize in the ER. Cells also continuously remove and degrade insulin receptors at the cell surface. Healthy cells maintain a constant number of insulin receptors at their surface by balancing their addition and removal. In obesity, accumulation of unfolded proteins in the ER causes ER stress. We found that ER stress inhibits the processing of insulin receptors in the ER. Transport of insulin receptors from the ER to the cell surface stops, but degradation continues. This balance of production and removal depletes the receptor at the cell surface, so the response of cells to insulin diminishes.
And at that point, you have insulin-resistant diabetes. You have fat cells that don't listen to insulin. Oh my God, does that get you into trouble because the fat cells can't take up energy anymore? You're going to starve? No, they're like doing that because they're completely full. They're chock full of all of that. So what's the downside of that? All that glucose in your bloodstream doesn't get taken up into target cells. And instead, it circulates around and around and around. It gets into trouble because it tends to clog up arteries, things of that sort. So, insulin-resistant diabetes, at that point, you could be secreting so much insulin it's dripping out of your nose, but if at the target end of things, the fat cells, if they have down-regulated their insulin receptors out of existence, it doesn't matter how much you're screaming with insulin. So, changing the number of receptors, the second half of the equation, how loudly you yell, how well you're being listened to. Another domain where you see this is receptors. Your average receptor is incredibly complicated. It's got different domains, different proteins, and it's coupled to an ion channel if it is a neurotransmitter receptor. If it is a steroid hormone receptor, it's got the hormone binding domain and the DNA binding domain. All of these are big, complicated molecules. Term in the business is they are made of protein complexes. There's 5 or 6 different proteins that combine to form this receptor. And 5, 6 different proteins, 5, 6 different genes. And you can get switching, subunit switching, where you'll take out this version of this receptor protein and replace it with this other version, which makes it a little bit more excitable or a little bit less so, or who knows what. You've got subunit switching there, which can happen in all sorts of circumstances. And there's a whole class of receptors for neurotransmitters in the brain, NMDA receptors, where depending on the composition, the subunit composition of these NMDA receptors, that influences how readily neurons learn new stuff. It modulates learning and memory. Okay, so another way of things being more complicated, the receptor is not the receptor monolithic thing. We just saw its numbers can change, it could increase, decrease, and its composition can be altered to change the flavor of its excitability. And you see the same thing in endocrine systems. Back to our example, before those fat cells are like totally fed up with trying to take on more glucose because they have no more room and they say, forget it, we're going to take away our insulin receptors. Before they do that, the first thing they do is they switch around some of the subunit features of the insulin receptor to make it less responsive to insulin. Same deal, change a little bit of the makeup of the receptor, but if that doesn't solve the problem and you're still being inundated with glucose in the bloodstream and you're still full, then you say, forget it, that's not enough. We're going to get rid of the receptor entirely. But the point there being, You change the number of receptors, increase or decrease them. You change the subunit compositions of them in different circumstances to make them a little more of this, a little bit less of that. And we can see all sorts of gradualist evolutionary change going on there in the genes coding for these subunits. Okay, so that's important. Next domain, more complicated than the dogma one gets in the introductions, which is this notion that here's the receptor for whatever, and it binds whatever. And that's the entire world of that receptor. It's the whatever binding receptor, and that's all you need to know. But then, following our similar themes here of multiplicity, you get all sorts of cases where receptors bind multiple things. Jargon in the field, multiple ligands. Do not panic at that word, not relevant, but where receptors bind multiple different things with interesting consequences. Example from the nervous system, neurotransmitter GABA. GABA, if you only need to remember one thing about GABA, if you're new to this business, GABA is inhibitory. It's the most inhibitory neurotransmitter in the brain. It is ubiquitous. It's all over the place. That diagram the other day of those feedback loops between the prefrontal cortex and the nucleus accumbens and the ventral tegmentum and all the little feedback loops that make us capable of being made happy by both symphonies and back rubs or something. All that feedback specialty is with little GABA releasing neurons that are inhibitory, just fine tuning the various loops. So GABA is an inhibitory neurotransmitter. Do not freak out at this phrase, but what it mediates is hyperpolarization of the neuron, making it less excitable. So we've got our GABA receptor here, and then along comes GABA from the presynaptic neuron, which is suddenly up and down instead of left and right. GABA comes in here, and it makes this neuron less excitable, less likely to have an action potential. So people had a fine time figuring out the molecular structure of the GABA receptor and all its different subunits, and then people began to notice that there were little side pockets on the receptors that bound other stuff. The first class that was identified were barbiturates. Barbiturates. What are they? They tranquilize you so radically that you become unconscious. Barbiturates, and as used for surgical anesthesia. What they do is they make the receptor much more responsive to GABA. So this inhibitory GABA signal, when there's barbiturates around, are so inhibitory that you're unconscious before you know it. So modulation at this major tranquilizer barbiturate binding site. What that suggests is, from that term, that there's a minor tranquilizing binding site also. And we've heard about these. These are the benzodiazepines. The muscle relaxants if you've got a muscle spasm, the relaxing your psyche if you've got anxiety. Benzodiazepines like Valium, Librium, the way they work to reduce anxiety, to reduce muscle tone. We heard the other day in terms of that James Lang negative feedback loop. Whoa, I still feel totally anxious, but wow, my muscles are so loose. I must not be feeling that anxious after all. That feedback business due to benzodiazepines, and they've got their own binding site. On the GABA receptor, it is not as potent of an interaction as with barbiturates, thus minor tranquilizers, major tranquilizers. So that was totally cool. People figuring out, wow, these two very important classes of drugs. There was a time that benzodiazepines like Valium were the most prescribed drugs in the United States. And this was a major advance. Oh, okay. So benzodiazepines are making the GABA receptor more sensitive to GABA and thus more hyperpolarizing, more inhibitory. And then people got their biggest surprise. They found a third binding site on the GABA receptor for the hormone progesterone. Okay, you should have all sorts of like bells going off in your heads at this point. bell number one. Wait, this is a hormone. We're talking about neurochemistry, neurotransmitters here. A hormone having an effect on the neurotransmitter receptor system? Yeah. Progesterone is a steroid hormone. We already learned what it does is it binds progesterone and then goes to a promoter. So it's doing stuff at the genes. What's it's doing? Having its own little binding site on GABA for progesterone and progesterone-like hormones, some of which are now known as neurosteroids. What they do is progesterone has a small amplifying effect on GABA, just like benzodiazepines. Progesterone, well, it's progestational. It's a hormone that has to do with pregnancy and all of that. Progesterone levels soar during pregnancy, and there's good reason to think that it has some strongly anti-anxiety effects by way of this pathway. And you better bet we will be getting soon to the whole business about mood changes, affect, negative affect around the time of one's period in a substantial subset of women, or in the aftermath of giving birth in the form of increased risk of a postparturition depression. What you see there, those are circumstances where progesterone levels are crashing way fast, and there's reasons to think that GABA is a little bit less effective as a result, and things like anxiety, depression risk become more prevalent there. So main thing there is, look at this. What is this gene coding genes coding for this complex of subunits? It's all about responding to GABA being modulated by these drugs, anesthetics, anti-anxiety drugs, progesterone with a bunch of behavioral effects we'll be learning about. So that's plenty interesting. How about multiple bonding sites at the hormone end? We just looked at a hormone interaction. Back to glucocorticoids, my beloved stress hormones bind to the glucocorticoid receptor. And what we saw was that that exposes a DNA binding domain, which binds to a promoter, and you turn on various genes. And there was a puzzle that emerged a while back that a number of labs, including mine, tried to sort out, which was, okay, stress, glucocorticoids, what do they do in the hippocampus? They screw up learning and memory. Glucocorticoids during major stress, they cause hippocampal neurons to atrophy and synapses to become less excitable, and that's when you're under stress, so learning is not ideal for you. So that's terrific. But then meanwhile, right next door on the amygdala, right next door, just the amygdalofugal pathway away, right next door in the amygdala, what does stress and lots of glucocorticoids do? It makes amygdala better at learning stuff, better at learning to be afraid of things. Amygdaloid neurons grow new projections during stress. They get more excitable. The amygdala gets bigger, while the hippocampus actually shrinks in some realms that we'll hear about. And what's the result? This is an amygdala that learns to be afraid of things that other people are not. This is the neurochemistry of why stress increases the risk of anxiety. So that's totally cool, all of that. But wait a second. The same hormone binding to the same receptor, which interacts with the same promoter in the DNA and in the hippocampus, it causes neurons to shrivel. And in the amygdala, it causes neurons to grow new projections. How could this be? This was utterly confusing to until it was figured out. Oh, there's a minor binding site for what is now termed a cofactor, a hormone receptor cofactor. And this one is present in the hippocampus, but not in the amygdala. And when this one is binding in here, X does whatever to the genome. And when this isn't here, X does exactly the opposite. So you're getting magnified ability to sort of regulate this whole system as a result of this cofactor stuff. So that's a whole additional world of hormones. And that explains things like why stress and glucocorticoids decrease inflammation in some parts of the brain and body, while increasing inflammation in other parts, something turning out to be very relevant to the inflammation in long COVID that goes on. So, what does this neurotransmitter do? Well, it's modulated by all these other bind. What does this hormone do? Well, it depends on whether this thing is around or not. So more complicated binding stuff there. Now, what this should begin to sound like to you is one of our themes from previous lectures. Well, what's the effect of glucocorticoids on expression of a gene having to do with neurons growing or atrophy? And the answer is, it depends. It depends. It depends. Are we in the hippocampus or the amygdala? It depends. Is this thing present or not? How inhibitory is GABA as a neurotransmitter? It depends. Are there benzodiazepines around? Are there barbiturates on board? What about progesterone levels? Conditional clauses. Modulatory ones straight out of our gene-environment interactions. If you are not able to say, ooh, what do glucocorticoids do to dendritic whatevers, if your answer has to be, it depends. It depends. Are we in the hippocampus or the amygdala? We are looking at an interaction, just like gene-environment interaction. This one is a hormone by brain location interaction, or a hormone by steroid receptor cofactor interaction. We know this concept. And these are more examples of the same. Here's a great example of it. So we've got neuron A, and what it does is it excites neuron B by way of releasing its neurotransmitter, which is blue, and binding to the blue receptor, and that's all great. And we see as part of this circuit, there's neuron C. Neuron C is doing totally nutty, crazy things. When you have been trained in like Neuro 101, the first bizarre thing about it is it's not sending its projections to the dendrites on another neuron. It's sending its projection to the axon terminal of this neuron. How weird is that? What, is going to release neurotransmitter there? Like there's going to be like receptors on the axon terminal for some other neurotransmitter? Yeah, that's the next bizarre thing. There's receptors here for the neurotransmitter coming out of here. We've already seen dogma violated the other day, presynaptic autoreceptors sitting there that could measure the amount of neurotransmitter being talked. But now we're seeing on axons, axon terminals, receptors for some completely different neurotransmitter system. This is as crazy as the other day postsynaptic neurons making and releasing neurotransmitters that go backwards, retrograde signaling. So here we've got this weird thing, and what we have is what is called an axonic synapse. So this has just broken all of our rules. To begin to get more of a sense as to what's going on here, neuron C releases GABA. This is a classic local little inhibitory neuron. What happens when GABA comes out and binds to its receptor, it's being modulated by all this stuff, binds to there, it silences, it hyperpolarizes, it makes less excitable this little stretch of membrane. What are the consequences of that? So this neuron is having an action potential and it's shooting down, and it shoots down here just fine and releases its neurotransmitters here. But because this GABAergic neuron has hyperpolarized this little stretch, it kills the action potential. It snuffs it out, and thus this neuron's action potential is unable to propagate and cause the release of neurotransmitters. So we have GABA blocking the excitation of B by A. So we can ask the same sort of question as before, which is, so now looking at this, what are the effects of GABA on neuron B? And what we know is, all on its own, GABA, neuron C, has no effect on neuron B. It has no direct effect at all. It influences this universe if and only if A is trying to talk to B. It does not play a causative role. It is modulating the means by which A talks to B. And it's our same exact deal again, which is, well, so what are the effects of neuron C on neuron B? And the answer is, it depends. It depends. If and only if A is trying to talk to B, then neuron C is really disrupting stuff. In the absence of it, it doesn't matter. Similarly, so now in the endocrine version of it, we've got that hormone CRH, which we saw comes out of the hypothalamus during stress and goes to the pituitary and releases ACTH. Why such a bizarre system is that? The whole separate hormone system with a whole separate little circulatory system. Once again, evolution isn't essentially efficient. It comes up, it improvises with stuff. But we have that system from the other day, CRH released during stress, and then causing the pituitary to do its thing through those corticotropes. And what we see here is if you add in that other hypothalamic hormone that's relevant, vasopressin, and add in vasopressin, put vasopressin by itself, it has no effect at all on ACTH release. But if you add in vasopressin when there's already CRH around, it greatly potentiates the effects of CRH. Well, what are the effects of vasopressin on ACTH release from the pituitary? And the answer is, it depends. It depends on its own. It does nothing. But if there's CRH around, it will greatly potentiate the response, modulatory. And what we should be able to do by now, what you should be able to do effortlessly at this point, is let's not even erase this diagram. We can say, well, what are the effects of this particular gene on growth of this plant? What are the effects of this gene? And the answer is, it depends. Are we talking about this much rainfall in the Sahara or this much rainfall in the Congo? A gene-environment interaction. We ask another one. Whoa, so what are the effects of having this gene, FAD-S2, on IQ, as we saw the other day? The answer is, it depends. It has no effect at all in individuals who were not breastfed. But if you were, it has a boosting effect. This is our modulatory theme. Over and over, what's the effect of having this bad news version of a gene involved in aggression we will hear much about? And the answer is it has no effect at all in the normal childhood, and only when it is coupled with childhood abuse. This is our thing. What's the effect of having two X chromosomes on your math skills? And the answer is, it depends. What sort of culture are you living in? If you are treated in a crappy, repressive way because you were female, it's going to have, like, math scores are going to be very different than if you're basking in the Icelandic sun's equality and all. Our theme over and over and over, whether gene-environment interactions, whether hormone neurotransmitter interactions, whether circuitry, whether all of these things, is over and over this theme of modulation. Modulation, what's the effects of this on that? It depends in what environment, in what hormonal background, in what genetic background. Modulation, our theme endlessly. So notice now we've got a double version of this hierarchically. Well, what are the effects of GABA on this neuron? And we've just seen that, well, it depends. It depends on if this neuron is trying to talk to this neuron. And if this neuron is trying to talk, C indirectly affects B. How much does GABA coming out of C affect the neuron under this circumstance? And it depends. It depends. Are there barbiturates? How much progesterone? How much benzodiazepines? All of that. Conditional clauses, modulatory, if then, if and only if these are around, GABA has a maximal effect there. If and only if that's happening in this synapse, if and only if this neuron is sending an action potential, then it has consequences here, hierarchies of our if-then clauses. By the way, you should be asking a question that comes to mind every time you're looking at how some drug does something to something in the brain, some neurotransmitter, whatever, and you'll say, well, the GABA receptor did not evolve in order to respond to somebody taking barbiturates or Valium, or what it tells in all those cases is lurking around in your brain is some endogenous barbiturate that binds there, and that's what it evolved for. There's some sort of endogenous benzodiazepine, and people have literally been looking for that for 40 years without much success. When you see a drug doing something or other, even a drug working here, this did not evolve for responding to that in drug. It tells you something or other inside the brain there resembles that drug, and go look for it. Okay, so we've had our big overview here as to all the ways the nervous system is not as simple as the dogma of introductory stuff. Likewise, the endocrine system, all sorts of ways in which there's more control, more regulation, more feedback regulation, more multiplicity of signaling with combinations, permutations that can have the same message here, but be doing very different things at this end, and thus getting specificity out of that. That being one big theme. Another big theme being modulation. Over and over, whether within this lecture, we are seeing a neurotransmitter, a part of the brain, a hormone, which is doing something if and only if it is coupled with something else. Modulation, interaction, the same thing out of all of our gene and behavior stuff. This is a theme, not just for the lecture, but for the entire course. You should be noting by now. Final key thing that comes out of all of this is look at the vast playground of possibilities here by which experience will change things, will change how much of this negative feedback loop is activated and how much of this is made, what alternative splicing of this or that, all the stuff we know already in terms of how dramatically things can change. And what is implicit in that is over and over and over helping to explain how you became you and why you is a very different proposition than whoever is sitting next to you. Individual differences sculpted by one's biological and environmental past.